Good morning, good afternoon, good evening to all the audiences. The goal of this presentation is to provide a high-level overview of CMC consideration for developing an effective mRNA LNP therapeutic. So the way I'll start is I'll focus on this word therapeutic. What exactly does it mean? Because mRNA LNPs traditionally so far have been successful in the vaccine space. Well, there's a lot of work going on in the therapeutic space, and then we'll focus on some of the CMC consideration. Before I go deeper into this, these are some of the disclosures, and I'll add one more qualifier in this. Throughout this presentation, when I use the word I actually mean we, because there is a team of scientists and engineers who are working on it. And if I use the word we, I actually mean they, because we have partners and collaborators who have contributed to the success of this program thus far. The way I have outlined this talk is the way I think of product development. So in terms of product development, I think of three C's that feed into your clinical and non-clinical studies. The first is your compelling biology. You can always have, even though we say we are modality agnostic, you can have success if you are focused on a compelling biology that will bring you success. So if biology is strong, the foundation is strong, everything else flows. So in this case, we'll use glycogen storage disease type 3 as an example. The second C on this is your CMC considerations. This for mRNA LNP includes formulating the LNPs, maximizing their shelf life, and minimizing reactogenicity. The third C, and that's how I'll sum it up, is your customer centricity. What does it take to be successful if you have these three C's? What are some of the high-level consideration? Now, one caveat with this is when you start product development, always start with the patient in mind in the jurisdiction, because there may be additional activities that may be required, but a high level overview is presented here. So when it comes to genetic and rare disease, here's an example of some of the current facts about it. So there are 350 million patients worldwide. That translates into 4% of the world population. There are 7,000 rare disease. 5% only have approved drug. 95% of these rare disease do not have a single approved drug. Of the 350 million, 30 million patients are in US, 50% are children. One is a big number if you have context for that one person. So if you think about it, on the individual cases, you may say these are rare, in the sense, by definition, a rare disease means 1 in 200,000 patient in US and 1 in 20,000 in European Union. But on a cumulative basis, the impact is vast. The second thing to note is, for many of these patients, the diagnosis by itself is a milestone. And lack of a therapeutic option means that prognosis is unchanged. A couple of weeks back, we had an ultra rare event where you invite the families and patient. There was a patient that was there who was diagnosed. It took her 46 years to be diagnosed with the disease. So glycogen storage disease type 3 is one such disease. It's one such rare disease, which is an autosomal recessive disorder caused by mutation in AGL gene. This gene, AGL gene, encodes for an enzyme called glycogen debranching enzyme, shown as GDE. Lack of or defect in GDE leads to glycogen accumulation, and the key symptoms are liver cirrhosis, enlarged liver cirrhosis, hypoglycemia, growth impairment, and dietary management tailored to individual patient using uncooked cornstarch remains the primary therapeutic option on the table. So Ultragenyx as a company has products which are very modality agnostic, from small molecule biologics, mRNA, LNPs to AAVs. And thus, when GSD3 solutions were thought about, all options were considered, starting with, if it's an enzyme replacement therapy, can we use a protein? The problem with using a protein is there is no intracellular trafficking. The problem with gene therapy provides one single permanent single dose solution, which is an upside. The challenge here is the limited packaging capacity of AAV precludes their use for delivering of gene greater than 4.7 kilobases. Now, this AGL gene is a 85 kilobase, contains 35 exons, and spans a mRNA that's 7 kilobases, thereby making mRNA as the ideal choice, because not only can you titrate the dose, but now you don't have to worry about the anti-GD gene therapy vector antibodies. So mRNA LNP was selected as the modality for treatment options or developing a product, and that's called UX053. And shown above this line is what happens in the absence of 053. There is a defect in GDE, and this defect in GDE is not able to process the glycogen. And what that leads to, it breaks some of these bound glucose, but you have branch glycogen. This is called a limit dextrin, which is an abnormal form of glycogen that keeps accumulating in your liver and muscle. If you have UX053, which is an investigational product, it consists of mRNA encoding a full-length GDE. GDE is, through its transferase and glycosidase activities, able to debranch. So the name says it all, glycogen debranching enzyme. So it debranches this glycogen by breaking alpha-1,4 and 1,6 glycosidic bond, thereby allowing glycogen phosphorylase to further break it and ultimately leading to release of glucose in the bloodstream. So there is a compelling biology to resolve this disease and develop some therapeutic option and restore the metabolism of limit dextrin. The challenge is when you think of mRNA LNPs for therapeutics, they are very different from that of vaccines. So the commonality is what is known as you have to bring to market safe. So you have this criteria, it should be safe, effective, and quality product, right? As listed here. And you should have ID, purity, content, potency. Those things have to be characterized and well defined within the regulatory constraint. That's a commonality. But the difference is there are finite differences between there are finite differences between a vaccine and the therapeutic, even though the modality is mRNA LNP. A successful therapeutic, you have to maintain the potency while minimizing immunogenicity. This is in contrast to vaccine. The dose, the dosing regimen, the durabilities are different. So if you look at the current COVID vaccine available here, they are 3,200 microgram, which translates into 0.1 to 0.4 microgram per kg. Durability wise, we have taken, most of us have gone through 2 doses followed by some boosters and IM route. In contrast, in a therapeutic regimen, your dose could be 100 to 1,000 fold higher. There may be need for more frequent dosing. And the other thing is vaccines are high volume product. And thus your scalability and the customer in the market segment is completely different. All that translates from a CMC perspective is your PAF is different. So by PAF, I mean the process, analytical, and formulation challenges are different. So throughout the remainder of the talk, we'll look at some of these aspects, starting with how do you maintain potency while minimizing immunogenicity. So the first thing on that one is, if you have to do this, you have to design the mRNA to reduce immunostimulation from the mRNA and increase the translation efficiency. If you look at the structural element of mRNA, the 5 moving from left to right, the cap, 5' UTR, ORF, 3' and poly A, some of the changes introduced are shown here. But in essence, what you're doing is you're taking your codon optimized and nucleotide modified mRNA. In our case, we screen in HepG2 cells for mRNA and protein expression. An example is shown here. What is shown in this orange box are all the genes and cytokines that are induced by the positive control. And when you do screen selection, the subset that are induced are shown by this yellow text. So and this was for sequence 3, which was further optimized. So that's one consideration, how you design the mRNA and selection of mRNA sequence. The second is the lipids. In our case, it's a proprietary lipid. The lipid that's selected is designed for rapid elimination using your PK studies, biodistribution. So you have to have a lipid that gets metabolized for with ease of elimination. So with that in mind, besides immunogenicity, so this is one factor, right? We start here, then we move into LNP process and formulation, and underlying all this is always going to be an analytical characterization. So besides immunogenicity, the other challenges intrinsic to mRNA LNP platform are its susceptibility to degradation, to enzymatic degradation, and almost negligible cellular uptake of naked mRNA. LNP, as we all know, they serve as the most advanced approach for encapsulating, protecting, and delivering this. However, up until this point, we were talking about the biology. Now we are focused on the chemistry and eventually to the physics. So from a chemistry perspective, what happens is you have all your lipid mixture in a polar protic solvent like ethanol, mRNA at low pH condition in the aqueous solution. When you mix those two, when the lipid meets the aqueous phase, it becomes protonated. It becomes protonated at pH 5.5, which is the halfway between the pK of the ionizable lipid and of the buffer, binds to the phosphate backbone, thereby experiences hydrophobicity, and that drives vesicle formation. And I have driven this vesicle like this as a small vesicle. So there are two things that are happening in this process. One, the prime interaction is the enthalpic interaction between this protonated ionizable lipid in the negatively charged phosphate backbone. The second is the hydrophobicity, because you're changing the solvent polarity in this process and binding them. As you further raise the pH in the dilution step, that neutralizes the lipid even more, increases its hydrophobicity, drives the vesicle formation together. And it's according to one of the publications from Dr. Bushman, late Dr. Bushman, 36 vesicles come together to form a LNP. And then PEG lipid forms the hydrophilic layer around it with DSPC bilayer underneath it. So understanding this, so the reason I'm talking about this in great detail, despite having so many publications, because these are self-assembled, multi-component complex system. It is the interaction of the LNP with the biological system that drives your efficacy in reactogenicity, and understanding them, designing them, controlling them becomes a key parameter to control your intended TPP or realization of your TPP for a given product. This is then followed by the form fill process. In an early phase, you can be a freeze-thaw stable formulation, or you have to then worry about your in-use and storage stability, followed by, in some case, like we were discussing before with the previous presentation, this could be a liquid state or a live state, and you have to then worry about tech transfer, scale up, and scale out consideration. So with this, the next few slides will focus now on the chemistry, eventually the physics of mixing, right? So in terms of starting pH condition, that was one of the questions last time. When shown on this slide is the structure and the stability. So on the left-hand side is shown as a circular dichroism of mRNA at pH 4 and pH 7.4. Circular dichroism, for those of you not familiar, you pass left, and this is what is shown by this blue and red. You pass left and right polarized light through a sample. If you have a chiral center, it will differentially absorb left versus right and give you a spectroscopic signature. So on the y-axis is your CD in millidegrees and in wavelength on x-axis. A double minima at 210 and 295 shows A-form helix with a band at 265. So at pH 7.4, you have formed more structure compared to a pH 4. So thus, if you start at a lower pH, not only it facilitates the positive charge for the ionizable lipid to facilitate binding, but it also keeps your mRNA in the elongated state. Similarly, when you think from a stability viewpoint, there's DSC that's shown on the right hand side. So heat capacity is on the y-axis, and the melting temperature is on the x-axis. mRNA in one of the buffers is shown by this green trace with a TM of 37C. You change the buffer condition, you get substantial stabilization as shown by this right shift of TM. But once you form the complex, if you do mRNA LNP DSC, it forms a single phase transition. That means these mRNA and lipids are coupled upon complex chain. So what that means is there's a very delicate and a fine balance between the formulation components, be it your buffer state or your pH in the processing condition. The practical impact of this is how your time in solution and your processing condition during scale up and scale out makes either you have purity or potency retention or loss. So the example here is if you look at DSC, even though TM is around 37C, you have onset of degradation much at room temperature, in which shows up. So even though that's an accelerated condition, in a practical setting, it shows purity on the left-hand side, in vitro potency on the right-hand side at 25C as a function of incubation on days. There is a linear loss of purity by CELF as well as in vitro potency, which is retained at 5C. So these parameters in determinations and hold times in temperature time variables, they become important when you're doing your process design space of what can be scalable. Similarly, if you think of encapsulation efficiency as a function of mRNA concentration, the buffer concentration in time that's shown here. So in all three graphs here, encapsulation efficiency is on y-axis, and in the first one, mRNA concentration is on x-axis. So encapsulation efficiency goes up in this condition as a function of mRNA concentration. What that means is if there is better mixing and better binding, encapsulation can go up during the mixing process. That was the PFT, the process flow diagram that was shown a couple of slides back. Similarly, if you have dilution buffer concentration, if you increase the buffer concentration, encapsulation efficiency could go down. This is because now you're shielding the charge. There is charge shielding, and thereby preventing the binding of lipid and decreasing the encapsulation efficiency. The surprising element here is stabilization time post dilution 1. If you hold it for a certain duration, encapsulation efficiency goes up. And that's why I was saying these are self-assembled multi-component unit, and there may be some more rearrangement that may be occurring in real time, be it fusing of those vesicles, fusing of LNPs, or encapsulation or binding of mRNA. And therefore, you will see encapsulation efficiency also goes up as a function of hold time. Besides the chemistry, so now we are moving from chemistry to the physics of mixing. And we looked at both T mixing and microfluidic mixing as a means of making these particle assemblies. And microfluidic mixing led to more uniform particle with a lower PDI, and that translated into a slight increase in vitro potency. Now, I do want to clarify, both T mixing and microfluidic mixing work well. What happens in the mixing processes, the particle assemblies are highly sensitive to the mixing process itself. At the collision between the jets, so when you have the ethanol and the aqueous phase coming together, the collision between the jet creates energy dissipation in the impingement zone. Greater is the energy dissipation, more is the mixing. So if you increase the flow rate or the Reynolds number, you'll see particle size goes down to a certain extent before it stabilizes. Now in microfluidic mixing, as most of you know, under the laminar flow, the two solutions will never mix together, but there are microscopic channels that are built into it that allows intermingling of these fluids on a very microscopic time scale, but in a very reproducible and a controllable manner. So both of them are known to work, and they have worked in our labs. The one you use is the one that fits your therapeutic or vaccine need, and that's where this comment is: scale up or scale out is a function of your market segment, the volume. So if vaccine is high volume, and I have a T mixer available, maybe that works. If I have a low dose situation and I don't want to lose a lot of product during my R&D, and this is scalable for my needs, microfluidic equally works as well. And so with this, we were able to take batch from as low as 30 milligrams to a 2-gram batch that's shown here. Considerations that went into it, the mixing process, and of course the equipment that comes with it, the shear associated with TFF, and that was captured last time, the buffer volume requirement, processing in whole times, which impacts your purity, potency, and things that were shown. So size, PDI, encapsulation efficiency, purity, and in vitro potency, they were comparable as we went from almost a 60x improvement. One more thing that was observed is illustrated here: stability during TFF. That purity is shown on the y-axis, and if you have a shear rate of 2500 per second versus 1500, at 2500 we did see some loss of purity during the TFF process. So your molecular weight cutoff, the type of membrane, the loading, the TMPs, all those things are considerations when you are scaling these batches up, besides your mixing process itself. So from once you have the batch made at different scale, the next thing you will be concerned about is your in-use stability and storage stability. From an in-use stability, as mentioned before, vaccines, mostly IM administered. Some vaccines are sub-Q administered. So from an in-use stability, in our case, it's an IV delivery. And what we found is if you use saline as diluent, your size may partially go up and you may have certain loss of encapsulation efficiency. Custom diluent may allow a new avenue for maintaining your stability during the in-use condition. Similarly, from a storage condition, and that was another, LIO is, like it was said before, LIO is right now getting a lot of focus and emphasis. It's because of the customer convenience it brings and allows getting vaccines to places where it's needed the most. With the current Moderna and Pfizer vaccine requiring their stability in frozen state as shown here, recently, now when I use the word recently, I think it's this month sometime, Gemcovac 19, there's a vaccine that was that received emergency use authorization. It's a self-amplified mRNA that's lyophilized and 228C stable in India. There are companies here who have gone into phase 3 with their lyophilized mRNA vaccine. So lyophilization is a very feasible option if you want to formulate for stability. It does add a unit up. So again, depending on a case-by-case basis, does it really fit the paradigm of the product you are trying to bring to the market? One may consider lyophilization as a add-on. So in our case, we used a 96-well lyo as a screening tool, and that allows us to preserve some of these mRNA LNPs instead of doing vial drawing. And what this shows is in formulations A1 through A5 and B1 through B6, we saw comparable performance of opalescence pre and post lyo condition. But in all cases, including all the publications I have seen to this date, there's a slight increase in hydrodynamic diameter compared to a pre-lyo control. Does it translate into a bioperformance issue? Yet to be seen. But given there is now an approved vaccine, even though it's emergency use authorization in a specific part of the world, that also facilitates its precedence and it's a doable option. So bringing all these things together in summarizing in the end, where I started from was always keep patient in mind in the jurisdiction. So what you want to get out the door is safe, effective, and quality product. And the place to start is the biology, starting with mRNA, LNP process in composition. So again, think from a biology to a CMC to a customer convenience. In this case, on the left-hand side, what I'm showing is go from biology to chemistry to physics. Once you have those, the next step is how are you formulating? Is it a liquid state or a lyo state? If a lyo, then look into freezing, fast freeze versus slow freeze, primary drying, gas transition below or below collapse temperature, secondary drying, the moisture condition, the storage, what type of diluent or container closure you will have, and stabilizing excipient. With all this, analytical is the underlying statement that includes your purity, your potency, your content, your uniformity, ID, all those become the underlining sections for product characterization. And once you have everything defined, then the biggest thing one has to consider, especially for mRNAs and LNPs, given their susceptibility to degradation, is raw material attributes, your equipment capabilities, and how those link to your process parameters, eventually to the drug product CQA, to bring to market safe, effective, and quality product. Um, with this, I'll pause. I do want to acknowledge the various team members who have contributed endlessly to this program in advancement of this platform at Ultragenyx. I'll be happy to take any questions. Thank you.